simple and complex patterns of inheritance. Okay. So mostly I want to just discuss the lecture questions, or sorry, the multiple choice questions for this topic. So the first one is number 20. So the TRP operon of E. coli controls expression of 5 enzymes that are involved in the biosynthesis of the amino acid tryptophan. The TRP repressor protein, which is the tryptophan repressor, binds to the TRP operator to prevent transcription when tryptophan is present. When would the tryptophan synthesis enzymes be made if there was a mutation in the DNA that prevented the repressor protein from being made? Right, the enzymes will be made in the presence or absence of tryptophan. So remember, for this operon, the only thing that's regulating it is that repressor. And if the repressor is not bound to the operator, then this operon is on. So if you have a situation where you never make the repressor, that means the repressor is never going to be bound, and that means the operon is always going to be on, regardless of whether you have tryptophan present or not in the environment. Okay, 21, the lack operon is expressed when? Right, C, great. So when the glucose is low and lactose is present. 22, when in a complex with blank, the cap protein binds to the cap site and blanks the expression of the lack operon. Right, D. So when in a complex with cyclic AMP, the cap protein will bind to the cap site and it's going to switch on the expression of the lack operon. 23, in the absence of lactose, the lack repressor is what? Right, active and can bind to the operator. Okay, the next few questions are about the TRP operon. 24, in the absence of tryptophan, the TRP repressor is not C. Right, inactive and cannot bind to the operator. So C is not right because it doesn't bind to the promoter. So yeah, you're right. It's E. It's inactive and cannot bind to the operator. 25, in the presence of tryptophan, tryptophan binds to the what? Right, E, the tryptophan repressor. And then 26, in the presence of tryptophan, the transcription of the TRP operon is on. Right, B, false. So remember that operon is only going to be on in the absence of tryptophan. Okay. Do you guys see any other questions that you want me to go through in terms of this topic? Okay. So let's move on to simple and complex patterns of inheritance. And for this topic, there's a lot of vocabulary that we have to go through. And you were already introduced to most of it in the pre-lectures, but we'll obviously go through it again in lecture. And what we're going to start off with when we study simple and complex patterns of inheritance is we're going to look at experiments done by Gregor Mendel using pea plants and how that led to some of the important things that we know about inheritance. When Mendel was studying inheritance, he used garden peas. And then, so the first thing we're going to go through is why did he use peas versus a different type of organism, different types of organisms that he could have used. And so one reason that he used peas in terms of studying heredity is that there were a lot of different kinds of peas that were available with easily distinguishable traits that can be quantified. Okay, And so easily distinguishable makes them easy to count. And then you can actually count them. So just as an example, these are the different characters. So you have a variety of pea plants that have purple flowers, but then there's another variety that has white flowers. And then they're either purple or white. Okay, so when Mendel was choosing his characters that he looked at, he chose to look at characters that were present in two easily distinguishable forms. So you have purple versus white. He also looked at flower position, axial and terminal, easily distinguishable, easy to count. And then he also looked at seed color. You have yellow seeds versus green seeds. So with every character that he looked at, he chose to look at characters that were present in two forms, and then he chose to look at, that were present in two forms, and that were easily distinguishable from each other. Okay, so this just shows you the different characters that he's going to look at. Backing up to this slide, also when you're doing genetic experiments, you need large numbers for your experiment in terms of getting statistically significant results. And peas are useful for that because they produce a large number of offspring, so you're going to have a large number of offspring to count in terms of whether they've inherited something or not. Also, they're small, easy to grow, and that makes them easy to work with. When you're studying the genetics of certain organisms, when you study the genetics of mice, that's going to take longer because they don't reproduce as fast as peas do. Another reason we looked at it was because he was actually able to manipulate the reproductive organisms so that he could control which pea plant is being crossed with which pea plant. So obviously that's not something you could do with humans ethically. You can't just decide which two humans you want to mate to produce offspring. And so it was important that you be able to do that, and then also that it's easy to do that. So it's easy to control which two plants you're trying to cross or mate with each other. And then it's also important that they were able to self-fertilize. The reason that it was important that they can self-fertilize is that we're going to see that when we look at his experiments, he began, and we'll define what this is in a second, he began with true breeding varieties of garden peas, and he produced those true breeding garden varieties through several rounds of self-fertilization. So the self-fertilization is how he's going to produce these true breeding varieties of garden peas. Okay. Did I take those out? I did. Okay. I'm not going to ask you to define this on an exam or quiz, but I'll just tell you what it means to be true breeding. So in terms of pea plants, if a plant is true breeding for a particular trait, that means that when you allow it to self-fertilize and you do that over several generations, you only produce that trait, which means that if I tell you have a plant that's true breeding for purple flowers, that means if you allow that plant to self-fertilize, you're always going to get purple flowered plants. And if you allow all those offspring to self-fertilize, you're only going to get purple flowered plants. And the same thing for white. If you're true breeding for white, all you ever get are white flowers. Okay, so that's what it means to be true breeding. And then, sorry, just to back up, to make sure, self-fertilization is when the two gametes that come together during fertilization come from the same individual. So meaning the sperm and egg both come from the same plant.
If they can self-fertilize, how do you make sure they weren't when you were trying to cross
them? In terms of controlling, so you would specifically take pollen from one.
They don't just do it naturally.
No, they do it naturally. They can do it naturally. Yeah. So, but he would specifically do the mating, and then those would be the offspring that he looked at. Okay. In terms of each of these things, the first three things that we want to discuss is, the first one we've already discussed, an allele, genotype, and phenotype, because these are just words that we're going to use a lot when describing the experiments. So remember that an allele is one variant form of a gene. Okay, so an allele is one variant form of a gene. So when we studied mitosis and meiosis, we said that in many cases genes come in multiple forms, and the multiple forms of a gene are called alleles. And then specifically, we'll see in this experiment that for some genes, you're going to have one allele that's called the dominant allele, and the other allele that's called the recessive allele, but not all alleles for genes are like that. They're not all dominant versus recessive. Okay, so genotype is the combination of alleles that an individual has for a particular gene. Okay, so the genotype is specifically the combination of alleles that a person has for a particular gene. So just backing up, we said that in some cases for alleles, you're going to have a dominant allele and a recessive allele. And when you have that as the case, you're going to say the dominant allele is the capital letter, and then the recessive allele is going to be a lowercase letter. So whenever you see a gene being defined as a capital versus a lowercase, that means that it's dominant versus recessive. Okay. Now, so remember that a particular individual is going to have two copies of every gene because they inherited one copy from mom and one copy from dad. So if I tell you that an individual is AA, that means they have two copies of the dominant allele. I'm telling you their genotype. So AA is a genotype. Also, A, lowercase a is a genotype, and then the third variety would be lowercase a. Those are all genotypes. So I'm telling you the combination of alleles that the individual has for that particular gene. Okay, and then phenotype is any observable characteristic of an organism. Okay, so the phenotype is any observable characteristic of an organism. And the relationship between genotype and phenotype is that the genotype determines the phenotype. Okay, so an individual's combination of alleles that it has for a particular gene is going to determine the observable characteristics of that organism. Okay, is everyone okay with those terms? And then also it's important to realize the association between genotype and phenotype. And again, genotype determines phenotype. Okay. So the next two useful vocabulary words we're going to go through is homozygous and heterozygous. And so a homozygous means that the two copies, the two alleles that you have for a particular gene are identical. Okay, so if you're homozygous, the two copies of the allele that you have for a particular gene are identical. So with respect to this example that I was writing up here on the board, individuals that are capital A are homozygous, and individuals that are lowercase a are also homozygous because they have two identical copies of a particular allele. And then to be more specific, we would call this homozygous dominant because it has two copies of the dominant allele. And then this would be homozygous recessive. Okay, and an individual that has two alleles that are not identical to each other, so has two different alleles, then you refer to that individual as heterozygous. Okay, so if the two alleles are different from each other, then that individual is heterozygous. So the person who has capital A, lowercase a, would be considered heterozygous with respect to this gene. Okay, any questions about homozygous versus heterozygous? And then in addition to that, homozygous dominant versus homozygous recessive. Okay. And then, so the word dominant can refer to an allele, and it can also refer to a trait, and the same thing for recessive. So a dominant allele is the allele whose phenotypic effect is seen in heterozygous individuals. Okay, so the dominant allele is the allele whose phenotypic effect is seen in the heterozygous individuals. So what that means is, let's say that A causes you to be, let's say, tall, and lowercase a causes you to be short. Okay, so the phenotypic effect of A is to be tall, and the phenotypic effect of lowercase a is that you're short. And heterozygous individuals have one copy of each of those alleles, and then the question is, which phenotypic effect do you see in that individual? And for this particular example, we'll say that they're tall. And so that means that the capital A allele, that's why you labeled it capital A, is the dominant allele, and the lowercase a is the recessive allele. Okay, so lowercase a would be the recessive allele, and then the definition for that is that's the allele whose phenotypic effect is not seen in the heterozygous individual. Okay, so the recessive allele, again, is the allele whose phenotypic effect is not seen in the heterozygous individual. So in this example, you do not see, it's not short, so you don't see it, and that would be the recessive allele then. Okay, and then dominant trait and recessive trait are pretty similar. So the dominant trait is the trait that's observed in the heterozygous individual. So with respect to tall and short, those are the traits. So tall is dominant to short because that's the phenotype you observe in the heterozygous individual. And then a recessive trait would be the trait that's masked or hidden in a heterozygous individual. So obviously because the individual is not short, that's the one that's masked or hidden, and that's the recessive trait. Okay, and then again, just to add this in there, not all alleles are dominant versus recessive. That's only true for some genes. The same thing, so then you're not always going to have a dominant versus recessive trait when you're talking about inheritance. Okay, this is just one pattern of inheritance can give you dominant traits versus recessive traits. So we're only talking about one pattern in this case. Okay, and then this is useful vocabulary for doing crosses. And so this is the different generations that you're going to have in a cross. So the first cross that you do in a genetics cross, so you have two parents, that's going to be the parental generation, and that's the P generation. Okay, so let's say I start with someone who is AA, and I'll cross them. So this means that I'm mating these two individuals, someone who's AA with someone who's lowercase a. And that's going to be my P generation. That's the first cross that I do. And then my first cross is going to produce a certain number of offspring. And the offspring of this cross make up the F1 generation. And F1 stands for first filial. First filial. Yeah. I was saying it incorrectly for a lot of years because I was adding in an extra O. So F1 generation is first filial, and the F1 generation is the offspring of the P generation. And then what happens in a cross, so in the crosses that we're going to see, you're either going to let the F1 generation self-fertilize, or you're going to take two F1 individuals and cross them, and then that's going to produce a certain number of offspring, and those offspring represent or are part of the F2 generation, which just stands for second filial. Okay, so this is just useful for knowing the different generations in a genetic cross. So when you're setting up an experiment, you can have a P generation, an F1 generation, an F2, and conversely, you can have some experiments that will have an F3 generation. Okay. Now the through is what a monohybrid cross and what a dihybrid cross is. So one thing that you want to keep in mind in terms of the difference is in a monohybrid cross, you're following the inheritance of only one character. So that's where the mono comes from. So a monohybrid cross is when you're following the inheritance of one character, and then you're following the inheritance of two different characters at the same time. Okay, so a monohybrid cross, again, is when you're following the inheritance of two characters, and it's going to happen when you cross two individuals that are heterozygous with respect to the gene that you're looking at. Okay, so the hybrid part comes from the fact that this individual here is a hybrid. They have a capital A and a lowercase a. And then you're taking two hybrids and you're crossing them. But you're only following the inheritance of this single gene A. Okay, so in a monohybrid cross, you're taking two heterozygous individuals and you're crossing them. So you can either think of it as in a monohybrid cross as you're following inheritance of one trait, or you can think of it as you're following the inheritance of one gene. Okay, and then in a dihybrid cross, you're following the inheritance of two traits or two genes. In a dihybrid cross, the individuals are heterozygous with respect to both genes. So you're looking at, let's say, A, but then you're also looking at gene B, and this individual would be heterozygous with respect to both genes. And then if you cross two individuals like that, that's going to be a dihybrid cross, because you're taking two dihybrids and you're crossing them. But for this class, you just mostly want to focus that in monohybrid crosses, you're following the inheritance of one gene or one character, and in dihybrid crosses, we're going to be following the inheritance of two genes or two characters. Okay. So the first thing that we're going to do is discuss Mendel's monohybrid crosses. And these monohybrid crosses are going to lead to what's called the law of segregation. Yeah, I'll say it later. So we're going to go through Mendel's monohybrid crosses first. And again, these crosses are going to lead to what's now called the law of segregation. So what I'm going to do is explain his experimental approach in these crosses, and then explain the results that he got. So basically what he did in these experiments is in the P generation, and he looked at several different characters. So in this, we're only looking at one character, which is plant height. Okay, so we're only following the inheritance of one character, plant height, and plants are either tall or dwarf. Okay, so in these sets of experiments, he took a plant that was true breeding for one form of the character. So he took a plant that was true breeding for tall and crossed it with a plant that was true breeding for dwarf. Okay, so, or true breeding for the other character. And then that cross is going to produce your offspring, which make up the F1 generation. Okay, then what he did is he allowed all the individuals in the F1 generation to self-fertilize, and that produced your F2 generation. And then what he did in each generation is counted the number of plants that he saw with each trait. Okay, and so let's just take what he saw in this example. So in the P generation, he crossed the plant that was true breeding for tall and a plant that was true breeding for dwarf. All the offspring in this generation were tall. And so from that, he concluded that tall is dominant to dwarf. Okay, so tall is the dominant trait and dwarf is the recessive trait. Then what he did is he let all these offspring self-fertilize. That produced the F2 generation. And he counted the number of tall and dwarf offspring. And in the F2 generation, he got approximately a 3-to-1 ratio. Another way of saying that is that approximately 75% of the offspring were tall and approximately 25% were dwarf. Okay, so essentially what he saw in this experiment is that in the F1 generation, he saw one of the traits disappear, and the trait that disappeared is going to be the recessive trait. and then that trait reappeared in the F2 generation, and then when you count the number of individuals with a particular trait, he got a ratio of about 3 to 1. Okay, and he saw this result with every character that he looked at. Okay, so this shows you the table that includes all the other characters, and so this would be the P generation cross that I did. So we took a plant that was triggering for axial flowers versus a plant that's triggering for terminal flowers. In the F1 generation, all the plants had axial flowers, which means that axial is dominant to terminal. And then we allowed all the F1 individuals to self-fertilize. That produced the F2 generation. And he again counted the number of plants that had axial versus terminal flowers. And again, he saw a ratio that was approximately 3 to 1. And so we saw that pattern for all the crosses that he looked at. So we saw one trait disappear in the F1 generation, only to reappear in the F2 generation. And then when he looked within the F2 generation, he specifically saw this approximately 3 to 1 ratio for all the crosses that he did. Okay, so that was the data that he collected from those experiments. And so from that data, he had 3 important ideas. One is that traits can exist in 2 forms, dominant or recessive. As I've already told you, that's not always true. So there are traits that aren't dominant versus recessive. So we'll see some examples of that. But they can exist in 2 forms, one being dominant, one being recessive. A second important idea that he had was that an individual carries 2 genes for a given character, and genes have variant forms, which we now call alleles. And then the third important idea, this is basically one way of stating the law of segregation, is that the 2 alleles of a gene separate during gamete formation so that each sperm and egg receives only 1 allele. Okay, so that's the law of segregation. Another way of stating the law of segregation is that it states that the 2 alleles in a pair separate from each other during gamete formation. Into separate gametes. I'm going to say into different gametes. Okay, so this is the law of segregation. So it states that the 2 alleles in a pair are going to separate from each other, I forgot to put the other in there, each other during gamete formation into different gametes. Okay, and this is related to, we'll see later on, I'll show you pictures, this is related to what happens during meiosis. So let's say that you're an individual that's capital A, lowercase a, and remember that the germ cells of this individual are going to be diploid, so they have 2 copies of every gene. But then when this cell undergoes meiosis, a particular gamete is only going to get 1 copy of this gene. So it's either going to get, it's either going to get a capital A, or the gamete's going to get a lowercase a, and that's going to happen during meiosis. And so that's what this is, that's when this is happening. So you're separating A from lowercase a during gamete formation, and a particular gamete is only going to carry 1 allele for that gene. Does that make sense to people? Okay. I'll have a picture of that later on to just provide you with a visual for that. Okay, and so this was just a slide that I put in there so that you could write the law of segregation on that slide. I should have said that before writing it out. Okay. So this is talking about the connection between the law of segregation and meiosis, which I just said, but this will provide you with, oops, nope. I am going to draw a picture. Law of segregation being connected to meiosis. So let's say that And in this case, you're following inheritance of one gene. So you start off with a cell, and the cell has two copies of this chromosome, and this chromosome carries the capital A allele, and this chromosome carries the lowercase a allele. Okay, and then remember, before you enter meiosis, you're going to replicate both those chromosomes. So that's going to produce a cell that has both the chromosomes replicated. And so this chromosome over here, both chromatids carry a capital A allele, and over here, both chromatids carry a lowercase a allele. Okay, so this is just DNA replication happening, and then this cell will undergo meiosis 1, and remember during meiosis 1, you're going to separate these two homologous chromosomes from each other. So you're going to produce a cell, two cells. One cell has the chromosome that consists of two sister chromatids, both of which carry the capital A allele. And then this cell over here is going to contain a chromosome that consists of two sister chromatids that carry the lowercase a allele. And then both of those cells will undergo meiosis 2, where you separate the sister chromatids from each other. And so this meiosis over here, this meiosis 2 over here, will produce two cells, and each cell will be a gamete that carries the capital A allele. And then this meiosis 2 over here will produce two cells, and both of these cells will have a chromosome in here that carries the lowercase a allele. So this connects the law of segregation with what happens during meiosis. This explains why they get separated from each other during gamete formation. Okay, is everyone okay with that connection? Okay, so what I'll do now is, because we won't make it through this with enough time, is we'll stop there. I'm going to pass back your exams.